Okay.
Okay, sir. And what is the constant feature of embryo sac?
What is the
Constant feature of embryo sac.
constant feature? Constant features means embryo sac has two poles. One is that of the Chalajal pole, other is the basically what is called microbial pole. In the microbial end, cells are present. In the middle, you will get the egg, and on both sides, you will get the synergies. And in the Chalajal pole, you will get three cells called antipoda cells. In the center of the central cell, two polar nuclei form, which may be united to form a secondary diploid nucleus. So this is what about the basic feature. Okay.
Okay, sir.
Now, generally, the embryo sac development in most cases is monosporic. I think you can remember what is monosporic development, right? You know this monosporic development? Ankon, do you have any idea? Monosporic?
Yes, sir. The method of embryo sac formation from a single megaspore.
Single functional megaspore. Okay. Now we have to study the hydrophily. It is not very common actually. Genera of angiospermic plants only. Most of them are monopods. Basically, water dependency is very rare in case of angiosperms because you know they are not to depend on water for the some formation of seed like that of algae, fungi, algae, bryophytes, etc. You know, for fertilization, water deficiency is now for fertilization, water dependency is found in algae, bryophytes, pteridophytes. Generally, in angiosperms is not found. So that's why it is only in 30 genera of the flowering plants you will get hydrophily. And another most important thing, it is not that in all aquatic plants hydrophily is found. There are a lot of aquatic plants where hydrophily is not found. Common one is water hyacinth. I think you know what is water hyacinth.
Sorrow Bengal.
And second one, pistia or water lily. And in there, you know, in them, you know, zoophily is found. Now, hydrophily is found in two types of plants are there. In Erisnaria hydrilla. The freshwater plants sea grasses like Zostera. Marine plants. Hydrophily is found. Velisnaria. In Velisnaria hydrilla, the freshwater plants, and in seagrasses like Zostera, you will get hydrophily. Now, what is the basic feature of hydrophily in them?
স্যার সাইডের জায়গাটা দেখা যাচ্ছে।
Sorry. Now you see, regarding now Velisnaria. Look one. This is the female flower with long stalk. Female flower with long stalk. These are surface of water. You can call it surface of water. Female flower reaches the surface of water, and male flowers, on the other hand, flowers are reaching there. So this is the male flower. Pollen grains are released on the surface of the water. And then they are carried to the stigma passively by water current. Carried to the female flower. Passively by water current. So this is what about the in Velisnaria it happens. So here the female flower reaches the surface with the help of its long stalk, and male flowers are detached from the marine body and carried to the surface of the water. Pollen grains are released onto the surface of water, and they are then carried to the female flower passively by water current. And some of them ultimately will reach the female flower and pollination takes place. This is what about the hydrophily in Valisnaria or in Hydrilla, the same. Next, in the other plant, can I rub this part? Right hand part, please. So this is the surface of water. And now you see the plants are submerged here. Both male plants and female plants are found to be submerged, and pollen grains are released from the anther, and passively by water current they are carried to the stigma of the female flower, and pollination occurs. This is pollination in Zostera. Is it clear now? The difference is clear? In case of Zostera, both male and female flowers are found submerged. And from the male flowers, pollen grains are released inside the water. and these pollen grains are then carried to the female flower passively by water current. This is what about the pollination in Zostera or sea grasses.
স্যার
Is clear? Huh?
নিচের জায়গাটা দেখা যাচ্ছিল না।
ঠিক আছে এবার?
হ্যাঁ স্যার।
কমপ্লিট কর। কোথেকে মেইন পাওয়ারটা এলো একটু দেখিয়ে দিই।
ঠিক আছে স্যার।
Now I have to rub it out. Complete it, please. General characteristics of hydrophilus flowers. Point 1. Flowers are small, without fragment, flowers the small, flowers do not produce nectar. Colorful. Sometimes in some cases ribbon shaped pollen grains are found. Ribbon shape pollen grains are found. Then you see lighter in weight,
Set ko rin tayo. Ito para sa tayo. Todo sila set ko din sini.
pollen grains are lighter in weight,
Basta mo. Sambulan. Ang pangbulan. Sorry. Ito ko rin po.
pollen grains are protected from wetting by mucilage covering.
প্রোটেক্টেড এর পর কি
Huh?
প্রোটেক্টেড এর পর কি এটা লাস্টের পয়েন্টে
Last one, pollen grains are protected from wetting by a mucilaginous covering. Covering. In most cases, male flowers get detached.
স্যার ওয়েল্টিং মানে
Eh?
ওয়েল্টিং মানে
Not wilting, wetting. ভিজে যাওয়া।
আচ্ছা ওয়েটিং এটা
The moment male flowers get detached, and in most cases, male flowers get detached from the plant and carried to the surface of water. The stigma of the female flower are with long coiled stalk. Stigma is there, like that. So these are the basic characteristics of
স্যার তুমি দুইবার 6 নাম্বার লিখে ফেলেছো
oh sorry. Okay. Now the general characteristics. There is nothing but the general characteristics of hydrophilous flower. Example already given. For the hydrophilous flower, example Zostera, Melisnaria, Hydrilla. Clear? I'm not going to name two aquatic plants where hydrophily is not found. The answer should be water hyacinth, water lily.
স্যার এটা 8 নম্বর হবে আর আগেরটা 7 নম্বর
Acha, okay. 1, 2, 3, 4, 5, 6, 7. Oh. 7. Then it should be 8. Okay. Now I have to rub it out.
Sir, 7 number point, what is male flatters?
In most cases, male flowers get detached. In most cases, male flowers get detached from the plant and carried to the surface of water. Already I have shown you in the diagram. Have you seen in the diagram?
Yes,
In
sir.
Belisnaria? In Belisnaria, what it is found, male flowers get detached. Are you getting me?
Yes, sir.
Okay.
Yes, sir. Now can I rub it out now? Yes, sir.
So remember this thing, both anemophilous flower and the hydrophilous flowers do not produce nectar, where they need not to attract insects for pollination. Are you getting me? So neither in anemophilous flower nor in hydrophilous flower, they are not colorful and also they do not produce the nectar. Actually, flowers are required to become colorful for attracting insects or animals so that they can help in the process of pollination. Now we are proceeding towards the next. Pollination by animals. This classification entomophily, Chiropterophily, Melacophily, etc. Entomophily, now pollination by insects, pollination by birds, pollination by bat and pollination by snail. Now we have to concentrate on further entomophilous flowers. Now here the pollinating agents. Bees, butterflies, fly, flies, beetles, etc. These are the animals which are taking part in the process of pollination. Moths. Now, first of all, we have to consider the general characteristics and then some special characteristics found in some special cases. Now, I have to wrap these three parts now: ornithophily, Chiropterophily, and Melacophily. Is it completed?
Yes, sir. Completely.
General characteristics of Entomophilous flowers. Entomophilous flowers, example, you know. Common examples: jasmine, sunflower, polyanthus, Amorphophilus, Musa, etc. Now, first of all, characteristics. Flowers are large, colorful, and rich in nectar. They are large, colorful, fragrant, and rich in nectar. Secondly, pollen grains are attractive for inviting insects. Attractive. Pollen grains are found sticky, then comes to the sticky and rubbing. Flowers are large in orchids. Different point, not general characteristics. So these are the general characteristics. Stigma is rough and at maturity stigmated fluid is secreted from it. So this is the general characteristics of the Entomophilous flower. Flowers are large, colorful, fragrant, and rich in nectar. Petals are found attractive and colorful. Pollen grains are sticky and rough. Stigma is rough and at maturity stigmatic fluid is secreted from it. And you know, they actually, the nectar is there to attract all these things. Now you see, this is the basic characteristics. Now we have to confine to some special characteristics found in case of different types of flowers. Now can I rub it out this part?
Yes, sir.
Yes, sir.
Okay. Now you see there are some flowers from where which are secreted at a foul odor to attract flies and beetles. To attract the flies. Is a special thing because normally flowers have to secrete sweet odor. Normally flowers are found to secrete sweet odor. In jasmine it is sweet odor. In some other flower, polyanthes, sweet odor. But there are some flowers where you will get foul odor. Flies and insects. This is one specialty. Another specialty you see. The interaction between flower and pollinators. So this is a flower. So what happens here? Pollinator animals visit flowers for pollens and nectar. And so pollinator animals visit flower for pollens and nectars. And in the meantime, they help in pollination. The flower providing them reward in the form of actually pollinator animals help in carrying out pollination in flowers. Flowers in turn give reward to them. So flowers in turn provide reward to these pollinator animals. And reward is nothing but the pollen grains and nectar. Whereas rewards are So animals have to visit flowers for consuming pollen grains and nectar, and while visiting different flowers, they help in pollination. And so that these animals can continue visit these flowers provide them with the reward, and here the reward is nothing but the pollen grains, and other is the other reward, the nectar. So this is the interaction. Is one form. Normally, what we have seen in the environment, that different animals are visiting flowers for nectar. And you know, if you have seen a lot of flowers are there around you, and a lot of animals are found visiting them. So this is the reward system. Here you need to remember this. This is also most important. But the reward may vary. In all cases, actually, in all cases, only the nectar and the pollen grains, not the rewards. Rewards may be found in the other form also. For example, in Amorphophilous flowers, the tallest flower. In Amorphophilous flowers, some moths are found to legs. So that means this flower provides safe place for, that is, these flowers provide safe place to the eggs, and this is the reward. In return, in this way, pollination occurs in these flowers. Look what happens here. Pollination animals help in carrying out pollination in flowers, and flowers in turn provide reward to these pollinator animals. So here, floral, actually, you know what happens in case of Amorphophilous, the floral reward is the place provide place for laying eggs. This is the reward here. So reward may vary. That's why I said earlier that in all cases the pollen grains and nectar may not be the reward. In case of Amorphophilous, the reward is to provide the safe place to lay eggs. Another condition is found in case of Eusa. This is the one first case. Eusa plant, the Eusa flower and moth. Eusa flower and moth, the next case. What happens here? I have to rub this part, please. This part only. Okay? Can I?
Yes, sir.
Okay.
Yes, sir.
Now you see, in this case, the moth is found to deposit its eggs in the locule of ovary of the flower. The moth is found to deposit its eggs in the locule of ovary of the flower. Benefit, what is the benefit? And the flower eventually gets pollinated. So here the pollen reward, floral reward is to provide space for laying eggs. That means so, and what happens then? Then the larva coming out during the development of the seed. Larvae of moth will come out when the seed starts developing. Begins. Larvae of moth will come out from where? From the egg. Come out of the egg when development of seeds begins. When development of seeds begins. So this is what about the, so here use of flower or use of plant and moth are interrelated. No one can complete his life cycle without the other. So this is what most important in case of use of flower and moth. Now these are 4 cases already discussed. Common case is the insect pollinated flowers are to release good smell or sweet smell to attract insects. In case of flies and beetle pollinated flowers, bad foul odor is found to be secreted. In case of Amorphophilus and also Usa, they are providing safe place for the actually laying of eggs or deposition of eggs. So in this way, the rewards may vary. Common floral rewards are always the nectar and the pollen grains. But there are some animals pollen robbers. There are some animals which are found consuming the pollen grains and the nectar, but they are not going to help in pollination. Flowers consuming pollens and nectar, but not going to help in pollination. These are called pollen robbers. Are you getting me? Who visit the flowers for consuming pollen grains and nectar, but not going to help in without bringing out pollination. Carpenter bees, bumblebees are the examples of pollen rovers. Clear is it up to this? Pollen rovers are actually consuming or stealing the pollens and nectar but without bringing about the pollination. Examples: carpenter bees, bumblebees. Now the plants are to develop some defense mechanism against them also. Some defense mechanisms, what are the defense mechanisms found? Can I wrap this part? Defense against pollen rovers. Defenses against pollen rovers. What are these? First of all, release of toxin and secondary compounds. And these are found effective only against the pollen rovers. Toxins and secondary compounds are found in nectar tissue, petal tissue. An interesting thing is that these compounds only affect the pollen rovers, not the true pollinators. Look here, release of toxin and secondary compounds. The toxin and secondary compounds are found in nectar and petal tissue. These compounds only affect the pollen rovers, not the true pollinators. Clear now?
স্যার
Yes.
স্যার ট্রু পলিনেটার্সকে কিভাবে এফেক্ট করে না ওদের কোনো
Look one thing. Actually, there is a time period. Generally, it is interesting phenomenon. There are certain times when actually pollen rovers are found to visit, but actual pollinators are not going to visit at that time. Are you getting me? So plants are to release this toxin at the time when pollen rovers are to visit. And the other time when pollinators are going to visit, suppose generally there are the flowers where pollinators are to visit at night. But pollen grabs are found to visit daytime. So during daytime, these flowers are going to release what? This toxin or secondary compounds. But at night, they are not going to release them. Are you getting me now?
Yes, sir.
Sometimes another thing is found. So actually, whenever pollen grabs have to visit the flowers, so flowers develop another method. What is found? So generally pollen grabs are found to visit this flower at noon, suppose. So these flowers are not going to produce any nectar at noon. Are you getting me? These flowers are not going to produce any nectar at noon. Rather, they are producing nectar at night or in the evening when true pollinators are to visit. In these also, actually, they can be protected from the pollen grabs. Are you getting me?
Yes, sir.
Okay. Now, this is all about the pollination. Now, I have to rub it out. Then comes to the outbreeding devices. Outbreeding devices are actually developed in plants to avoid self-pollination. And encourage cross pollination. Why? Because continued self-pollination results in inbreeding depression. Continued self-pollination results in inbreeding depression in plants.
So inbreeding depression ki?
If a plant is going to reproduce or where continuously self-pollination is going on, then you know the genes present in the plant are only carried to the offspring. So here, the chance of variation is very less. As you know, environment is regularly changing. If variations are not occurring regularly, then it will be very difficult for the plant to survive properly in this environment. So its productivity may be decreased, its efficiency decreases, and these all are the inbreeding depression. Are you getting me now?
Yes, sir.
স্যার এই ডিভাইসগুলি কিভাবে কাজ করে?
Yes, I am coming to this. I am coming to every device. Actually, all these devices are to encourage cross-pollination but to discourage the self-pollination. Because if self-pollination is discouraged, then this then the chance of inbreeding depression is not there. Okay. Now I am discussing one by one. First of all, pollen release and pollen release and stigma receptivity are not synchronized. That means either pollen grains are released earlier, then stigma become receptive, or pollen stigma becomes receptive much before then the pollen release. Two things happens. Either pollen grains are released from anther before the stigma become receptive, or so both these things happens. Suppose you know. So as I said earlier, in case of autogamy I discussed, the pollen release and stigma receptivity are required to be synchronized to facilitate self-pollination. So here you know, in this case what happens, the plants are evolved such technique that the pollen maturation and stigma maturation are not going to take place simultaneously. Either pollen grains are matured earlier or stigma becomes receptive earlier. So that why such changes are taking place? These changes are taking place to avoid self-pollination. So in this way, autogamy can be avoided. Autogamy can be avoided. Clear? Now the next one. Here of this part, right hand part.
Okay, sir.
Next. Anther and stigma are placed at different position. Are placed at different position. Look one thing. Look at this. So this is the anther and this is the stigma. So you know anther and stigma are found to be at different position, and this position cannot help in self-pollination. Is it clear? As anther and stigma are placed at different position, So this can't help in self, because what I said earlier, in order to ensure autogamy, the anther and stigma required to be present very close to each other. As I said earlier, in order to ensure autogamy, anther and stigma are required to be present very close to each other. But you see, here anther and stigma are found to be at different positions, and this position can't help in cell pollination. So, in this way, autogamy can be prevented. Now, third one. Self-incompatibility. Actually, you know, in order to avoid self-pollination, there is a genetic characteristics here actually, genetic mechanism in order to prevent the self-pollination. To prevent self-pollination. And what is this mechanism? I am showing you. Can I graph this part? The pollen release and stigma receptivity, point 1. Can I graph point 1?
Yes, sir.
Thank you. Look one. If similar self-incompatibility factor, listen carefully, if similar self-incompatibility factor is found in both anther and stigma, then although pollen grains are shifted to the stigma, but they will never germinate. I am repeating it again. If same self-incompatibility factor is found in both anther and stigma, then the pollen grains, after being carried to the stigma, will never germinate. Is it clear? Now I am showing you. Now you see, the self-incompatibility factor found in the stigma is S1 and S2. And here is also S1 and S2. So the pollen grain released one is S1 type, other is S2 type. When they are carried over here, a similar self-incompatibility factor is found in both, so these pollen grains will never germinate. Is it clear?
Sir, তার কি genetically different S1 এবং S2
Basically, S1 and S2, they are the self-incompatibility factor actually. If similar self-incompatibility factor is found in the anther and also in the stigma, then what happens? Then you see that these will prevent the germination of pollen grains. That means if it is found that here it is S1, pollen grain is also S1, then stigma is not stimulated to release the stigmatic fluid, or the chemical dialogue cannot be developed between these two to facilitate the release of stigmatic fluid. Until stigmatic fluid is released, the pollen grains can't start germinating. Are you getting me?
Yes, sir.
Now you see, I am showing you actually in other two cases. Then it will be better for you to understand. Can I rub this part, please? This part, right hand part.
Yes, sir.
Okay. Now you see what happens. I am showing another two diagrams here. Same stigma. Suppose S1, S2. Also S1, S2. Now pollen grains from other sources are coming like this. If S3 and S1 are landed onto the stigma, then here what happens? The S3 will germinate to form pollen tube. So you can call this condition Hub incompatible. Compatible. Now, if in that case, you can call it so if in a flower you have to concentrate on it, self incompatibility, not these two diagrams actually. So here you see in this way self incompatibility prevents autogamy. And J2-nogamy. Is it clear now? Because it is not only the same plant, plants are produced same incompatible form, the factor is found the same plant. So in other, that's why this mechanism can prevent both autogamy and J2-nogamy. Can I rub it out, please?
Yes, sir.
Is it completed? Is it understood? Okay. Can I rub it out? আমি তো একটু ওর মধ্যে ক্লাসে আছি, একটু কতক্ষণ করতেছি। Can I rub it out now?
Yes, sir.
Yes, sir.
So third one was self-incorporated, and fourth one is production of unisexual flowers. Hello? Production of unisexual flowers. Two types may be found. Female flower and male flower in the same plant. This is one condition. If male flower and female flowers are produced in the same plant, then this unisexuality, this type, can only prevent autogamy. This type can only prevent autogamy because here you see, as male and female flowers are found in the same plant, so J2-gamy can be occurred. Now other keys. This is plant A, two different plants. This is the male flower. Female flower. This type can prevent both autogamy and J2-gamy. That means it is found in example papaya. Cucurbita. So in cucurbita, male and female flowers are produced in the same plant. So this is also unisexual flowers. But as male and female flowers are found in the same plant, so autogamy can be prevented, but J2-gamy cannot be prevented. In this case, like papaya, male flowers are produced in one plant and female flowers are in the others. And in this case, both orthogamy and heterogamy can be prevented. So the chance of variation is more in the second case. So this is what about the production of unisexual flowers. So in this way, there are 4 outbreeding devices discussed here to make you understand that how these devices can help in discouraging self-pollination but encouraging cross-pollination. Clear? Is it clear to everybody?
Yes, sir.
Okay. Now, so our breeding device is completed. So you need to remember, because these questions are coming in board and also and from in entrance also the questions are coming from it. So you need to go through every line, whatever it is written. Now I am to rub it out. Completed?
Yes, sir.
So we can say one thing, dioecious condition helps in preventing both orthogamy and heterogamy. Explain. Can you explain this? Dioecious condition can help in preventing both orthogamy and heterogamy. I think you know dioecious means when male flowers are produced in one plant and female flowers are produced in the other plant. This case. Are you getting me? Dioecious conditions actually is helpful in preventing both orthogamy and heterogamy. Dioecious condition means when male flower and female flowers are found to be developed in 2 different plants. Clear? Is it okay?
Yes, sir.
Okay. Now next part is pollen-pistil interaction, and I discussed it in my next class. Okay. Pollen-pistil interaction. Okay.
Okay, sir.
Okay, prepare yourself. You need to complete this task by today, please. Everyone, I am requesting everyone to complete your task by today itself. Okay, I am going to wrap it out. Okay, or close this class, please. Any doubt? Any doubt? Also, there is no doubt. So I am going to. Okay, thank you. So we meet again on next Wednesday, next Monday. Thank you very much.